In the previous session, we have learned about isolation of plasmid DNA. Today's session we will be learning about cloning vectors, mainly focusing on plasmids. What is vector in biology? Vector means carrier. A mosquito is called a vector because it is carrier of many viruses and bacteria that causes diseases. In genetic engineering, likewise, vectors are carriers of desired gene fragment. DNA molecule to become a vector should satisfy certain criteria. It should be smaller in size, and it should have the capacity to self-replicate and be independent from the genome of the host. There are two main prokaryotic vectors which satisfy these conditions: plasmids and bacteriophages. Bacteriophages, although they don't be like a living organism outside a host, when they inject their genome into the host, the genome does not integrate with the bacterial genome and is self-replicating. What are plasmids? Plasmid, if you see the image, you can know they are circular, double-stranded, self-replicating, independent DNA. They are not integrated with the host chromosome. They are independent. Just by looking at its characteristics, you can list 3 or 4 characteristics of plasmid that forms the definition. Plasmids are known to carry one or more genes that are of special attribution to the plasmid. They are not necessary for the normal survival of bacteria, but they attribute special characteristics like antibiotic resistance or defense against other bacteria, are playing a major role in degrading certain organic chemicals, and likewise many other qualities. Antibiotic resistance. In the laboratory, antibiotic resistance is used as a selectable marker. Selectable marker is a gene which is inserted in the plasmid to know whether the plasmid is transferred into the host and it is undergoing a replication in the bacteria. To know the successful expression of that bacteria plasmid, like if a plasmid for antibiotic resistance is inserted in the bacteria with antibiotic resistance as a selectable marker, its expression in the bacteria is known through experiments, like with growing this bacteria in antibiotic prone media. If this bacteria is successful in growing, then the results confer the expression of this vector in the host. A plasmid possesses the origin of replication site. Since it is self-replicating, it must contain origin of replication site, and it is independent of the main chromosome. Although smaller plasmids are preferred, sometimes these smaller plasmids are using DNA replicative enzymes of the host, while the larger plasmids carry its own gene, and it produces special enzymes only required for plasmid replication. Here is a picture of the synthetic plasmids, that is PBR322 and PUC19. These two are generally used as selectable markers in DNA transfer experiments to ensure proper transfer of vector in the host chromosome. There are integrative and non-integrative plasmids. Non-integrative are the ones that do not integrate with the bacterial chromosome, while the integrative plasmids are the plasmids which integrate with the host chromosome and is known as episome. These may not remain integrated throughout the life of the bacteria. During certain cell divisions, these may be liberated and again form a separate unit known as plasmid, which is independent and self-replicating. Another factor that plays main role in genetic engineering is the copy number and the size of the plasmid. As we have known earlier, smaller plasmids are fit for cloning. But plasmids range from 1 kb to 250 kb size, but the plasmids required for cloning is just less than 10 kb. That's the reason only few are useful for cloning purposes. Under circumstances that are special, larger plasmids are also used in cloning purposes. The copy number refers to the number of molecules of an individual plasmid that are normally found in the single bacterial cell. Like if plasmid is required in large numbers, its amplification number plays a very important role. Like large number of plasmids is required to produce desired genes, inserted clones, to ensure maximum happening of the experiment. Plasmids can be conjugative or non-conjugative. Conjugative plasmids are the ones that can be transferred from one bacterium to another, while the non-conjugative plasmids cannot be transferred. These non-conjugative plasmids can be transferred with the help of conjugative plasmids sometimes. One of the characteristic features of F plasmid is it is a conjugative plasmid that helps transferring of non-conjugative plasmids. Classification of plasmids based on their function. F plasmid or fertility plasmid, resistance plasmid or R plasmid, call plasmids, and plasmids in plant-associated bacteria. It is the image stating the basic regions for plasmid, like a plasmid with origin of replication site, a promoter, restriction site where the gene of interest is inserted, a selectable marker where expression of that particular gene is studied, and the antibiotic resistance gene. F plasmid is the conjugative plasmid. The F plasmid contains genes called tragenes that helps in the synthesis of sex pili, which forms a bridge between two cells, like the donor cell and the recipient cell, and aids the transfer of plasmid. The transfer does not mean the plasmid, when transferred from donor to recipient, becomes absent in donor cell. When it is transferred, it is mostly one of the strands which is being transferred through rolling circle replication, and another strand is complementarily synthesized in the recipient cell. Hence the reason when the plasmid is transferred from donor to recipient, it still remains in the donor also. The next type of plasmid is the resistance plasmid. Resistance plasmid is a very important plasmid in the field of genetic engineering, as this is the plasmid in which natural antibiotic resistance genes are seen. Some plasmids may have different antibiotic resistance genes, like many plasmids may be present in a single cell with different resistance gene, like a plasmid for tetracycline resistance, plasmid for ampicillin resistance. Sometimes a single plasmid can have multiple gene resistant areas, like a single plasmid containing ampicillin gene resistant area or a tetracycline gene resistant area. And this resistant plasmid is conjugative. An important and interesting fact is the resistance plasmid need not be transferred between the species only. They can be transferred between wide range of bacteria and induce virulence in the transferred bacteria. The next type of plasmid is the call plasmid. Call plasmid is a type of plasmid that is present for bacterial immunity against other bacteria. The call plasmid is used to synthesize a bacterial toxin called colicin, which actually protects the host that produces colicin toxin and infects and kills. It actually kills the related bacteria by piercing its cell membrane and altering the osmotic pressure. This is actually having a great application in the field of food industry, where it is used as a food preservative. And also in recent days, researchers have been conducting various experiments to accommodate this in the field of antibiotics, to kill a broad range of antibiotics through the colicin toxins produced by this. Another type of plasmid is the degradative plasmid. This plasmid is also a very wonderful plasmid, as it can degrade natural compounds like the organophosphates, and other compounds that are not degraded in nature and are very harmful to nature after its usage when left in excess. These plasmids are evolved in such a way that these degrade this organic compounds that are hazardous in a very effective way, and this is employed in many biodegradation studies conducted in recent days. Another type of plasmid is the plasmid which induce virulence in plants. A bacterium called Agrobacterium tumefaciens carry genes that cause tumor-like infection in plants known as crown gall disease. Usually, this bacterium without the plasmid is actually harmless to the plant. It does not cause disease, but when TI plasmid is present, actually TI plasmid is formed due to a viral gene transfer through virulence. This TI plasmid, when present in the Agrobacterium tumefaciens, can cause disease. In its absence, the plant is safe. This method of gene transfer is actually exploited in biological method of gene transfer in plants, as a transfer of gene can be obtained by cutting off the virulence gene in this TI plasmid and inserting the gene of interest in this plasmid, and biological gene transfer methods can be achieved through this plasmid. Another example of plasmid-induced infection is the rhizobium. Rhizobium is a very popular bacterium in nitrogenous fixation. This bacterium actually establishes a symbiotic relationship with the plant and aid nitrogen fixation, but primarily this actually infects the plant because of the presence of plasmid. This infection lodges this bacterium into the plant, later establishes a symbiotic relationship with the plant. If you find this video informative, please do like, share and subscribe to our channel. If you want to know more about individual plasmids and methods of their transfer and infection, please do check for the link in the description. Thank you.